to Science Online. My name is Rajesh. This video is for A-level biology students, and the topic I'm going to discuss today is the dihybrid inheritance. At the end of this video, you should have the clear understanding of this inheritance and how to draw the Punnett square for different genotypes. So let's see. Before we go into the details, let's clarify a few terminologies. The first, genotype and phenotype. What is a genotype? Genotype is the genetic makeup of an organism. The phenotype is an observable characteristics of an organism. Say for example, black hair. The genes which are responsible to create the black hair is a genotype. The appearance of the character, which is the black hair, as a phenotype. The next, the dominant allele and the recessive allele. What is a dominant allele? It's an allele which expresses its character when it's homozygous dominant or heterozygous. We normally represent the dominant allele in uppercase letters. The recessive alleles, the allele can only express its character when they are homozygous recessive. Recessive alleles represented by lowercase letters. During fertilization, there are lots of genes passed from generation to generation. This is what we call inheritance. In this topic, we're only going to talk about two genes responsible for two different characters are passing from one generation to another. This process is what we call dihybrid inheritance. In this example, we're going to talk about two different characters in plants. The first one is the color of the seed. The dominant character here is the yellow color, which is represented by capital G. The recessive color is the green color, which is represented by small g. The second character is the shape of the seed. The round seed is the dominant, represented by capital R. The wrinkled seed, which is recessive character, represented by small r. Please note, these two genes are sitting on two different homologous pairs of the chromosomes. That's very important for this dihybrid inheritance. In this example, if you look at the parent's phenotype, the first parent is the round and yellow dominant character. The second parent is wrinkled and green recessive character. But if you look at the genotype of each parent, it will give you a little bit more information. The first parent is homozygous dominant. It's a pure breed dominant. Both characters are capital R, and capital G, capital G. Same goes to the second parent is a pure breed recessive. Both of them are small r, and small g, small g. Therefore, the only gamete the first parent can produce is capital R, capital G. And the second parent, small r, small g. These are the only two gametes can be formed by each parent. So during the fertilization, when these two gametes fertilize, in the F1 generation, the offspring is going to be round and yellow because there's a dominant characters are coming from one parent. So the genotype of the F1 generation is going to be capital R, capital G, small g, because we have the dominant characters are here. The phenotype of the offspring is going to be round and yellow. Now, if we use the offsprings of the first fertilization to carry out another fertilization, remember all the offspring of the first fertilization is heterozygous for both characters. So capital R, small r, capital G, small g. So these two parents are now going to create four different gametes since they are heterozygous for both characters. So the four different gametes, so capital R, capital G, capital R, small g, small r, capital G, small r, small g. Same from both parents. Since we have four different gametes, it's going to be lots of different combination, and therefore to work out the F2 generation, we need a Punnett square. Let's see how the Punnett square is going to be. I'm sure that you are familiar with Punnett squares. So let's write all the gametes for the Punnett square. So you write the gametes from one parent on one side and the other parent on the other side, and you use those combinations to fill the squares. Say for example, first one is going to be capital R, capital G gamete from each parent. Therefore, the first combination is capital R, capital G, capital G. So in this way, you can fill the whole Punnett squares with the possible combinations. Once you have done this, if you look at the phenotype of each of these possible combinations, you will see there are 9 round yellow seeds and 3 round green seeds and another 3 wrinkle and yellow seeds and only 1 wrinkle and green seeds. So this is the combination for this crossing. So in the F2 generation, we have the ratio of 9:3:3:1. So this is the dihybrid inheritance ratio. Please note, this is a theoretical ratio. In reality, this ratio can deviate from its values. There's a number of reasons for that. Independent assortments, crossing over, or even mutation can cause this change. The other important thing you need to note in genetics, the ratio we apply is at every single fertilization. It's not an overall ratio. Say for example, if you are tossing a coin, there's a 50-50 chance of getting a head and a tail. So this probability or the ratio is at every single toss. So if you are tossing a coin for say 4 times, there is a possibility you can still get heads on each 4 atoms. So this is the dihybrid inheritance and its ratio. In your exams, most of the time, they won't tell you what type of inheritance they are talking about, whether it's a dihybrid or something else. You should be able to figure out from the ratio or the outcome which inheritance is in the question. So it's very important to understand this process and the ratio and how we get to this ratio. Hope you find this video useful. Until I see you in another video.